So hello students and welcome back to yet another video tutorial. In today's video tutorial, we would be talking about the structure and the conformation of the DNA polymerase. That how does the DNA polymerase 1, which is also referred to as the Kornberg polymerase, catalyzes the biosynthesis of the DNA. Kornberg polymerase किस तरीके से DNA को बनाता है, आज हम ये देखने वाले हैं. इस लेक्चर के अंदर अभी आप देखेंगे that why does the Kornberg DNA polymerase is important and what are the key important activities associated with the Kornberg polymerase. So let us start with our lecture.
Genetic information to be passed on from a parent cell to a daughter cell, the parental DNA must be copied into two identical daughter DNA molecules.
See, it is the DNA molecule that is always transferring the information from the parents to the offspring. So in this manner, the parental DNA must be copied into the offspring DNA so that the information can be effectively transferred from the one generation to the next generation. For this process, the DNA is the genetic material which is present in our cell, and this process is being catalyzed with the help of an enzyme that is referred to as DNA polymerase. Now, how does this DNA polymerase act? That we will have to visualize.
You have completed this exercise. You should understand the chemistry of DNA synthesis, understand the mechanism of catalysis by DNA polymerase, and understand the proofreading function of DNA polymerase.
So in today's lecture, we would be talking about the three different things. One is understand the chemistry of DNA synthesis, that how does the chemical reactions are being involved in the process of DNA biosynthesis taking place. Understand the mechanism of catalysis by DNA polymerase, how does the catalysis takes place, and understand the proofreading function of the DNA polymerase, that in what manner the DNA polymerase catalyzes the proofreading of the newly synthesized DNA molecule.
DNA synthesis requires deoxynucleoside triphosphates and a primer template junction.
See, each and every DNA biosynthesis always requires certain nitrogenous bases which must be incorporated in the growing nucleotide chain. किसी भी डीएनए बायोसिंथेसिस के अंदर नाइट्रोजनस बेसिस को इनकॉर्पोरेट करना होता है ताकि वो नई बायोसिंथेसिस चेन को बना सके, एंड दीज नाइट्रोजनस बेसिस आर प्रेजेंट इन द फॉर्म ऑफ न्यूक्लियोटाइड्स. Nucleotides are present in the form of nucleotide triphosphate. So you can look here. So there are three phosphate molecules which are present. The innermost phosphate is commonly referred to as the alpha phosphate. The second phosphate is referred to as the beta phosphate, and the third phosphate is referred to as the gamma phosphate. So there are three different phosphate: alpha phosphate, beta phosphate, and the gamma phosphate, which are involved. And here you find that this nitrogenous base and the C1 carbon atom of the C1 prime carbon atom of the sugar molecule, that is referred to as the ribose sugar. And in the case of ribose sugar, it is beta D deoxyribose sugar, beta D deoxyribose sugar which is present. Okay, it is deoxyribose sugar which is present, and the C1 carbon atom hydroxy group is being bound to the nitrogen atom with the help of a bond, and that bond is commonly referred to as N-glycosidic bond. Okay, so this is N-glycosidic bond which is present. Another bond that is present here is the ester bond. There is yet another bond that is present, and this bond is commonly referred to as the ester bond. Okay, then in between the alpha, beta, and the gamma phosphate, there are bonds which are present, and these bonds are referred to as anhydride bonds. What are these bonds referred? These bonds are referred to as anhydride bonds. So these are all anhydride bonds, okay, that are present. So you can find an anhydride bond there and another anhydride bond here. So these are the different bonds which are present. These are the bonds which are present, and this is referred to as phosphoanhydride linkages. Okay, so these are the phosphoanhydride bonds. Phosphoanhydride bonds. So these are the phosphoanhydride bonds which are present, and it is these phosphoanhydride bonds that have the energy present within themselves. Whenever a nucleotide enters into the growing towards the 3' end of the growing primer region, then it always enters in the form of nucleotide triphosphate. जब भी न्यूक्लियोटाइड आता है किसकी फॉर्म में आता है? न्यूक्लियोटाइड ऑलवेज एंटर्स इन द फॉर्म ऑफ न्यूक्लियोटाइड ट्राइफॉस्फेट, एंड व्हाइल इट हैज बीन इनकॉर्पोरेटेड, only nucleotide monophosphate is going to be incorporated. The pyrophosphate is released in this process, that is PPI. Both the beta and the gamma phosphates are being removed in this process, and these phosphate molecules then further undergo degradation, leading to the liberation of large amount of energy. It will further degrade, leading to the formation of inorganic phosphate, two molecules of inorganic phosphate, and producing very large amount of energy in the entire process. So let us look at how this process of DNA polymerization takes place with the help of Kornberg DNA polymerase. कैसे Kornberg DNA polymerase इस पूरे DNA polymerization को कराता है, तो इसे अभी हम लोग देखते हैं.
Each of the four DNTPs, DGTP, DCTP, DATP, and DTTP, has three phosphoryl groups which are attached via the 5' hydroxyl of the 2' deoxyribose.
You always know that the purines and pyrimidine differs from each other on the basis of their junction with the sugar molecules. In the case of purines, there are two rings which are present, and the imidazole ring and ninth atom is always being bound. मैंने आप लोगों को क्लासेस में पहले भी बताया है, पहले की एनिमेशंस को आप देखेंगे, चार्ट को देखेंगे. It is the N9 nitrogen atom that is involved in the case of purine. Okay, and it is always the N1 nitrogen atom that is involved in the case of pyrimidines. N1 is involved in the case of pyrimidine. Okay, pyrimidine. So here you find that the nitrogenous bases which are present are adenine, guanine, cytosine, and thymine are the four different nitrogenous bases that are being involved in this entire procedure. Next point.
The phosphoryl group closest to the ribose is the alpha phosphate. The middle group is the beta phosphate, and the outermost group is the gamma phosphate.
So these are the three different phosphates which are present: alpha phosphate, beta phosphate, and gamma phosphate. One thing that you must always understand, that these phosphate molecules always have a negative charge. It is because of the negative charge of these phosphate molecules, because of the presence of negative charge of these phosphate molecules, that is why the DNA achieves a negative charge. That is why the DNA has a negative charge over itself, has a negative charge over itself, Why it has a negative charge? It has a negative charge over itself because of the presence of this phosphate molecule that has a negative charge. Okay, then the repulsion in between the two phosphate molecules are being minimized with the help of the magnesium ions which are present. Okay, so there are magnesium ions which are present in between the two phosphate molecules. Because of the presence of magnesium ion, the negative charge in between the two phosphate molecules can be minimized. Okay, this is how magnesium ions are being present. Okay, next point.
The primer template junction has two key components. The template provides the single-stranded DNA to be copied. The primer provides a free 3' hydroxyl at which a DNTP can be added.
Okay, so there is the primer template junction which is present. The template is going to be a single-stranded DNA which would be copied in the form of the daughter strand. Okay, this template will act as the parental strand, and there would be a newly synthesized strand that would be produced against it, okay, through the growing 3' hydroxyl group. तो यहाँ पर जो strand आपको दे रखा है वो template strand है, और template strand के सामने जो grow कर जाएगा that would be referred to as the replicating strand. Okay, here it would be the daughter strand that would be produced, or because of the activity of DNA polymerase, daughter strand that will be produced. Next point. So this is how the process is going to take place. Here the enzyme that is going to work is going to be referred to as the DNA polymerase. It is going to move here, and then it is going to add nucleotides towards the 3' 3' हाइड्रोक्सिल ग्रुप ऑफ द प्राइमर के 3' हाइड्रोक्सिल ग्रुप के ऊपर नए नए न्यूक्लियोटाइड, वेदर इट इज एडेनिन, गुआनिन, साइटोसिन, थाइमिन, दे विल बी एडेड on the basis of their hydrogen bonding on the complementary strand of the DNA. If there is thymine, adenine will be added. If there is cytosine, guanine will be added. If there is guanine, cytosine will be added. If there is again adenine, thymine, then here it is thymine, then here it would be adenine. So on the basis of the complementarity, the nucleotides are going to be added upon. Next.
The 3' hydroxyl of the primer strand attacks the alpha phosphoryl group of the incoming DNTP in a nucleophilic substitution reaction.
So there, the alpha phosphoryl group is going to be attacked. When the alpha phosphoryl group is going to be attacked, in the case of nucleotide substitution reaction, the beta and the gamma phosphate groups are going to be excluded. They are going to be removed. When this beta and the gamma phosphates are going to be removed, they will leave in the form of pyro-inorganic phosphates. So this would be called as pyro-inorganic phosphate, inorganic phosphate. Okay, and this pyro-inorganic phosphate is going to be lysed. When this is going to be lysed, it is going to free the beta phosphate and the gamma phosphate from each other. ये क्या करेगा? ये बीटा phosphate को और ये gamma phosphate को एक दूसरे से अलग कर देगा. Here it would be the gamma phosphate and the beta phosphate getting separated from each other. यहाँ पर ये beta phosphate है और ये दोनों एक दूसरे से separate हो गए. This separation or the hydrolysis of the pyro-inorganic phosphate is responsible for catalyzing the entire process of DNA replication. It is this pyro-inorganic phosphate hydrolysis that provides the energy. Hydrolysis provides the energy for DNA replication, the energy for effective DNA replication. For effective DNA replication. Okay, next point.
The leaving group for the reaction is pyrophosphate, which is formed from the beta and gamma phosphoryl groups of the DNTP.
Okay, you can call it as pyrophosphate or pyro-inorganic phosphate. It is the same thing, and that is going to be produced. Next.
The free energy change for this reaction is rather small. However, additional free energy is provided by the rapid hydrolysis of the pyrophosphate into two phosphate groups. This reaction is catalyzed.
Pyrophosphate के breakdown होने की वजह से ही large amount of energy is being provided. So it is very clearly mentioned here. Pyrophosphate hydrolysis by the pyrophosphate phosphatase enzyme is responsible for providing additional energy to take place. Okay, the additional energy is being provided by pyrophosphatase enzyme, which is going to break the pyrophosphate into two molecules of phosphate. When these two molecules of phosphate are going to be disintegrated, then they will provide the extra amount of energy that is necessarily required for the DNA replication to take place. तो यही दोनों excess amount of energy को provide करेंगे अच्छे से हो सके. Okay, so they would be providing the energy so that the effective reaction can take place. Next point.
By the enzyme pyrophosphatase.
Okay, so this is the major enzyme pyrophosphatase which is present. Moving to the next slide.
The process can then be repeated with the 3' hydroxyl of the newly added DNTP serving as the nucleophile.
So similarly, on the basis of the complementarity, new nucleotides are going to be added. कॉम्प्लीमेंटैरिटी के बेसिस पे दोनों के सामने नए नए न्यूक्लियोटाइड्स को आप ऐड करते चले जाएंगे. एट द प्लेस ऑफ साइटोसिन गुआनिन, एट द प्लेस ऑफ एडेनिन थाइमिन, गुआनिन साइटोसिन, थाइमिन एडेनिन. दिस इज हाउ द प्रोसेस इज गोइंग टू बी कंटिन्यूड.
The chemistry of DNA synthesis requires that DNA be made in a polar fashion. In biological polymerization, DNA is always made by extending the 3' end of the primer strand.
One thing which is very important here is that DNA is always synthesized in the 5' to 3' direction. When you say that how the DNA is going to be biosynthesized, then you can look here. DNA is going to be biosynthesized from the 5 prime towards the 3 prime direction. Okay, the arrow indicates towards the 3 prime. Okay, so this is how the DNA replication is going to take place from the 5 prime towards the 3 prime end of the DNA. Okay, so in this process the DNA replication is going to take place. Nucleotide triphosphate NTPs are going to come here, and NTPs are going to be incorporated in the form of nucleotide monophosphate with the liberation of pyrophosphate. Pyrophosphate are going to be broken down further, liberating large amount of energy. Okay, this is the gamma and this is the beta phosphate. They will liberate about a very large amount of energy. This is how the entire process is going to take place. Okay, and the DNA is going to be biosynthesized from the 5 prime end towards the 3 prime end, whether it is on the leading strand or on the lagging strand. चाहे लीडिंग स्ट्रैंड पे बायोसिंथेसिस हो चाहे लैगिंग पे, दोनों पे डीएनए की बायोसिंथेसिस हमेशा कहां पर होती है बेटा? 5 प्राइम एंड से 3 प्राइम एंड की तरफ. Now we would be looking at the molecular mechanism, that what is the mechanism of DNA replication and how does the mechanism takes place. Next point.
The template strand directs which of the 4 possible DNTPs is added. During replication,
during the process of replication, you can find that there would be wrong nucleotide triphosphate that would be coming, but the wrong nucleotide triphosphate are being repelled out. और जो सही वाला होता है ना, उसके तरफ अगर आप देखोगे, then that correct nucleotide will be held with the help of the bonding, the hydrogen bonding with the adjacent nucleotide, जिसको आपने थोड़ी देर और रोक लिया. अगर आपने जाते समय इस न्यूक्लियोटाइड को यहां पर डीएनए ने थोड़ी देर और रोक लिया तो वहीं पे बॉन्ड बन जाएगा. आपको ये पता होना चाहिए कि कौन सा न्यूक्लियोटाइड वहां पर जुड़ रहा है. अगर कॉम्प्लीमेंटैरिटी है, कंपैटिबल है तो रुक जाएगा, कंपैटिबल नहीं है तो इट्स ऑल ओवर, टाटा बाय बाय, एंड देन द न्यूक्लियोटाइड ट्राइफॉस्फेट्स विल बी रिमूव्ड. फॉरवर्ड.
The DNTP that base pairs with the template strand is highly favored for addition to the primer strand.
Okay, so the deoxynucleotide triphosphates are being added and the strand is going to be elongated. Next.
The main enzyme of DNA synthesis is DNA polymerase. The DNA substrate sits in a large cleft of the DNA polymerase that resembles a partially closed right hand. Based on the analogy to a hand, 3 domains of the polymerase called the palm, fingers, and thumb have been described.
So now, on the basis of the similarity of the DNA polymerase 1 with the hand, we can say that the DNA polymerase 1 is directly equivalent to the hand of a person. ओके, तो अगर आप अपने हाथ को देखें, ठीक है, तो फेयरवेल में आप किसी का हाथ मांग सकते थे, ठीक है, बाद में भी आपने किसी का हाथ मांग सकते हैं. सो व्हाट यू फाइंड हियर इज दैट this is the hand, and in this hand what you are going to find, there are fingers which are present. Okay, so there are fingers which are present, there is a thumb which is present. Okay, and then there would be a DNA molecule. Okay, let me take the DNA molecule as a particular wire. So I have a wire, so let this wire be a DNA molecule, and DNA molecule is going to bind over this particular thumb, over this particular hand. The thumb will be used to hold the DNA molecule. Okay, once it will be able to hold the DNA molecule, then it will hold the DNA molecule like this, and from here the melting of the DNA is going to take place. Once the melting of the DNA will take place, a single nucleotide will be free which can bind to the complementary incoming nucleotide. एक एक करके हम न्यूक्लियोटाइड्स को फ्री करेंगे और उनके सामने नए नए न्यूक्लियोटाइड्स को ऐड करते चले जाएंगे, जिससे हम डीएनए को पॉलीमराइज करा सके. नेक्स्ट पॉइंट.
The palm domain houses the active site for DNA synthesis.
Now see, there are 3 domains which are present in this hand. Which are the 3 domains? The palm domain, the thumb domain which is present. Okay, and the third one is referred to as the finger domain which is present. Okay, so the palm domain will have the active site where the process of DNA replication will take place. The thumb domain will hold the DNA molecule so that during the process of replication it does not slips from the active site, and these finger domains will cause the nucleotide triphosphate to come in close proximity with the DNA molecule so that it can bind. तो यहां पर अगर आप डीएनए पॉलीमरेज को अपने हैंड से कंपेयर करते हैं तो आपको 3 डोमेन मिलती हैं. कौन कौन सी? फिंगर डोमेन palm domain and thumb domain. First, palm domain. palm domain has a function. palm domain का जो काम है वो क्या है, डीएनए को एक्टिव साइट पे बाइंड करना. एक्सोन्यूक्लियस एक्टिविटी भी यहां पर प्रेजेंट होता है palm डोमेन के अंदर ही. थंब डोमेन का काम क्या है, डीएनए को केवल बाइंड करके रखना, डीएनए को बाइंड करना. और फिंगर डोमेन जो होती हैं वो एक्टिव साइट के लिए न्यूक्लियोटाइड ट्राइफॉस्फेट को एक्टिव साइट तक लेके आती हैं. कैसे लेके आती हैं वो हमें यहां पर देखना है.
The active site of DNA polymerase is able to distinguish between RNTPs and DNTPs, even though
Now, you might be thinking, sir, how to differentiate between the nucleotide triphosphate and deoxynucleotide triphosphate? The DNA polymerase itself has its active site designed in such a manner that it will only allow the deoxynucleotide triphosphate to bind. Active site में इतनी जगह ही नहीं है कि एक OH और एक्स्ट्रा आके राइबोन्यूक्लियोटाइड ट्राइफॉस्फेट के साथ में जुड़ सके. Deoxyribonucleotide triphosphate ही केवल जुड़ सकते हैं.
So RNTPs are present at approximately 10-fold higher concentration in the cell. The nucleotide binding pocket is too small to accommodate the presence of a 2' hydroxyl on the incoming nucleotide, allowing the polymerase to sterically exclude RNTPs.
So ribonucleotide triphosphate are sterically excluded from the active site of the enzyme, and only deoxynucleotide triphosphate is going to bind to the active site of the enzyme. Next point.
Correct base pairing is also required for catalysis. If an incorrect base pair forms, the alpha phosphoryl group of the DNTP cannot properly align with the 3' hydroxyl of the primer strand.
See, there was no complementarity between adenine and guanine, because adenine was trying to bind to guanine, but guanine was not able to effectively bind with the molecule. As there was no complementarity, so the bond was transient. It was just a passing affair. Okay. And so ultimately the guanine is going to be lost. And then what? Then do not get depressed. You will get a good thymine coming next after the guanine. एक बस जाती है तो दूसरी बस आती है. One nucleotide leaves, then only the second nucleotide is going to come and bind. So do not get depressed if one of the nucleotide has lost you. Just wait for the next nucleotide to come. So let us look here.
Once the proper DNTP is bound in the pocket, the reaction can continue.
So once the proper
palm
is going to be bounded, the reaction is going to continue. Okay. So now the previous DNTP which was present was not compatible. So we have removed it out, and then we have done an undo with that. Okay. So let us break that bond, the transient hydrogen bond, and let us make a new bond. And we have made a bond with the thymine. तो पहला वाला बॉन्ड सही नहीं था, अपन ने बाय बाय कर दिया, टाटा, इट्स ऑल ओवर बिटवीन द एडेनिन एंड गुआनिन. एंड सेकेंडली व्हेन द थायमीन came, we said yes, okay, it's good, it's thumbs up, and we have bounded the adenine with thymine because there was a high compatibility in between the two. Okay. Next, the phosphate molecules. Then there are certain phosphate molecules. अगर कभी भी आपकी लाइफ में कुछ अच्छा हो रहा है तो आपके छोटे भाई बहन जरूर होंगे. You will have certain brother and sisters who will spoil you every of your plan, or you will have your true friends who are going to spoil every of your plan. So remember to call certain magnesium ions for these phosphate molecules, because phosphate molecules can collide with each other and they will cause the nucleotide triphosphate to be dissociated into fragments. So that is why the magnesium ions will minimize the intramolecular repulsion in between the phosphate molecules. So there are two magnesium ions which are present, that is referred to as metalline A and metalline B, which are present in between the alpha beta and the beta gamma फॉस्फेट. तो मेटालाइन जो होते हैं अल्फा बीटा और बीटा गामा फॉस्फेट के बीच में, वो इंट्रामोलिकुलर रिपल्शन को मिनिमाइज कर देते हैं. नेक्स्ट पॉइंट.
Domain also binds two divalent metal ions. These metal ions are crucial for DNA polymerization activity.
So there are two metal ions, as I have told you, and these metal ions are essentially required for the DNA polymerization activity to take place.
Once a correct base pair is formed between the incoming DNTP and the template, the finger domain moves to enclose the DNTP. This conformational change
brings correct base pair बन गया, फिर तो कुछ नहीं है, फिर तो दो चार लोगों को बताना होता है, फिर उसके बाद में they will push you and there will be a permanent bond that will be produced. Okay, तो यहाँ पर एक correct base pair बनने की देरी थी. Once a correct base pair has been formed, once there has been a complementarity, then what is going to happen? The other guys are going to push you up and then they will make you form a permanent bond that is going to be produced, and this is being produced with the help of finger domain. Tathastu. Okay, so this is the thumb domain is used to bind to the DNA molecule. The DNA, it binds to the active site which is present over the palm, and then there is the finger domain, and this finger domain causes the nucleotides to come closer to each other, and then there is a fight in between the two molecules. Okay, that comes later. Probably someone knows in the class.
Brings the divalent metal ions into the correct positions to function.
Now the divalent metal ions are in the correct position to function, and the 3' hydroxy group is going to attack the innermost phosphate. It is going to form a phosphodiester linkage.
Metal ion A helps to deprotonate the 3' hydroxyl of the primer, producing an oxyanion to attack the alpha phosphate of the incoming DNTP.
So this oxyanion is going to attack the alpha phosphate of the incoming nucleotide triphosphate. It is going to attack the innermost alpha phosphate, and it is going to make the phosphodiester bond again, because this bond which was previously being produced, I am indicating it with my arrow. That one was a phosphoester bond. Another ester bond will be produced, so that would be referred to as phosphodiester bond. पहले भी एक ईस्टर बॉन्ड था फॉस्फेट और कार्बन के बीच में, सॉरी, अब फॉस्फेट और ऑक्सीजन के बीच में एक और ईस्टर हो जाएगा, तो यहां पर फॉस्फोडाइस्टर बॉन्ड्स विल बी प्रोड्यूस्ड. नेक्स्ट पॉइंट.
Metal ion B coordinates the negative charges of the beta and gamma phosphates of the DNTP,
yes,
and stabilizes the pyrophosphate leaving group. Lysine and arginine residues on the finger domain also help to stabilize the pyrophosphate. And
so pyrophosphates are being stabilized not only by the metal ions which are present, but the lysine and arginine groups which are also present over the finger domain are going to stabilize the pyrophosphate molecules, because lysine and arginine being positively charged, they will attract the negative charge towards themselves and will cause the stabilization over the negative charge phosphate molecule to take place. Next point.
And through stacking interactions, a tyrosine residue attached to the fingers helps to hold the DNTP in place for catalysis.
Now, this tyrosine is going to hold the deoxynucleotide triphosphate for the catalysis to take place. This tyrosine molecule is also present over the finger domain. Tyrosine molecule finger domain पे होता है, lysine finger domain पे होता है, arginine finger domain पे होता है. तीनों मिलके होल्ड करते हैं इनकमिंग न्यूक्लियोटाइड ट्राइफॉस्फेट को, एंड देन देयर इज अ रिएक्शन दैट इज गोइंग टू टेक प्लेस, एंड देन देयर इज अ फॉस्फोडाइस्टर बॉन्ड्स व्हिच आर गोइंग टू बी produced. So this is how the phosphodiester bonds are going to be produced. Next point.
The finger domain also plays a second role. The finger domain associates with the template region, leading to a nearly 90-degree turn of the phosphodiester backbone of the template immediately after the active site. This conformation of the template leaves only a single template base in the active site, preventing any confusion as to which base is ready to pair with the next nucleotide to be added.
So what you find here is that it is because of this finger domain that is responsible for the catalysis to take place. This finger domain makes a transient 90-degree turn within the DNA structure. As it makes a transient 90-degree turn within the structure of the DNA, it causes a single nucleotide to be exposed at the active site. And it is this nucleotide to which the incoming nucleotide triphosphate is going to bind. Once the incoming nucleotide triphosphate binds to it, then a new strand of the DNA is going to be biosynthesized. This is how process is going to take place. The 90-degree turn ensure that there is a single nucleotide that is going to be exposed in front, and this single nucleotide is going to be bounded with the incoming nucleotide triphosphates.
In contrast to the fingers and the palm, the thumb domain is not intimately involved in catalysis. Instead, the thumb interacts with the DNA that has been most recently synthesized. This serves to maintain the correct position of the primer template junction in the active site and reduce the rate of dissociation of the polymerase from the DNA substrate.
So it is the thumb domain which is going to hold the DNA molecule. Okay, so if there is a DNA molecule that is present, okay, let this be a DNA molecule that is present, and this DNA molecule is going to be held by the thumb molecule. Okay, so it is in this manner that DNA is going to be holded by thumb. Okay, Buddham Sharanam Gacchami. Okay, so you would have heard about Lord Buddha and his hand. His hand, he always used to bless the this. So this structure is that of DNA polymerase 1. He is going to tell you that how the process of DNA replication is going to take place. This is how the DNA is being held by the thumb. Okay, next point.
Another function of the palm domain is proofreading. The palm region makes extensive hydrogen bond contacts with the base pairs in the minor groove of the newly synthesized DNA.
देखिए, पाम डोमेन का एक और फंक्शन होता है. ये क्या करता है? ये प्रूफ रीडिंग को कराता है. कैसे प्रूफ रीडिंग को कराता है वो हमें यहाँ पर.
The palm region makes extensive hydrogen bond contacts with the base pairs in the minor groove of the newly synthesized DNA.
So we know that the DNA is present in the form of an helix with two different groups. One is referred to as the minor groove, another one is referred to as the major groove. DNA polymerase 1 has an ability to perform proofreading. Okay, and it performs the most extensive proofreading among all DNA polymerases. सारे के सारे डीएनए पॉलीमरेज में से सबसे ज्यादा इफेक्टिव प्रूफ रीडिंग अगर कोई करता है, then it is always the DNA polymerase 1 which performs the most effective proofreading among all DNA polymerases. So DNA Pol I performs the most effective DNA proofreading. Performs most effective DNA proofreading. Most effective DNA proofreading. So what is the proofreading that it performs? It performs the proofreading in both directions, 5' to 3' proofreading as well as 3' to 5' proofreading. These are the two different types of proofreading that are being performed by the DNA polymerase. DNA polymerase 1 दोनों ही तरीके की proofreading को perform यहाँ पर करता है. Next point.
These contacts are not base specific, but they only form if the recently added nucleotides are correctly base paired.
Okay, so only if the recently added nucleotide is correctly base paired, the DNA is going to bind to the minor groove. If the recently added nucleotide is not correctly base paired, the DNA will not bind to the minor groove. अगर सही तरीके से न्यूक्लियोटाइड जुड़ा है तभी डीएनए माइनर ग्रुप से बाइंड करेगा. सही तरीके से न्यूक्लियोटाइड आने वाला न्यूक्लियोटाइड नहीं जुड़ा है तो डीएनए माइनर ग्रुप से बाइंड नहीं करेगा और डीएनए विल बी एक्सक्लूडेड फ्रॉम द माइनर ग्रुप. Once the DNA excludes from the minor groove, DNA is being pushed towards the exonuclease site, which is also present over the palm domain. और exonuclease site पे जाके गलती को हम सुधार देंगे. अगर गलती से कोई मिस्टेक हो जाए तो डीएनए पॉलीमरेज क्या करता है? उस मिस्टेक को रिकॉग्नाइज करता है. ओके, इट वाज अ मिस्टेक जस्ट फ्रॉम माय साइड, इट वाज जस्ट अ मिस्टेक. तब मिस्टेक को करने के बाद में वो क्या करता है? अंडू कर देता है. वो बैक जाता है, उस मिस्टेक को रेक्टिफाई करता है और फिर नई मिस्टेक की तरफ, नई करेक्टेड मिस्टेक की तरफ फिर आगे बढ़ता चला जाता है. सो द थिंग दैट इज वेरी इंपॉर्टेंट इज दैट if there is a mistake that you have made, you can just rectify it out and then move forward. Looking at the animation.
In the rare event that a mismatched base pair is added, the distorted geometry of the substrate causes the replication rate to slow dramatically. The palm domain.
If a wrong nucleotide has been added, then a mismatch which has been created will cause the replication rate to be drastically reduced. When the replication rate is drastically reduced, then the DNA replication is going to slow down. As it is going to slow down, so DNA will be pushed towards the exonuclease site and the exonuclease site will remove the mismatch base pair. And then the process will again replicate and the DNA will be made to replicate.
Is now not able to make contacts with the minor groove. And consequently,
so the palm domain is not going to make contact with the minor group. Why? Because the palm domain has lost the contact because there was a mismatch base pair that has been introduced in the DNA. Once the mismatch base pair has been introduced, then the DNA is pushed towards the exonuclease site where the mismatch base pair would be executed. It will be removed off. गलती से जो मिस्टेक हुई थी उसे सुधारने के लिए हम एक्सोन्यूक्लियस साइट पे पहुंचे और जो मिस्टेक थी उसे हमने रिमूव कर दिया. नेक्स्ट,
primer template junction is free to move about and interact with the exonuclease site.
Okay, it is this exonuclease site where this nucleotide, wrongly incorporated nucleotide as shown in the yellow color, will be excised off.
The exonuclease site removes incorrectly base paired nucleotides starting from a 3' DNA end and is therefore called a proofreading exonuclease. Removal
so this is
of
the
the
proofreading exonuclease site, which is going to remove any wrong incorporated nucleotide towards the 3'.
Correctly paired base is a matter of kinetics. With a mismatched base, the diminished replication rate is now slower than the exonuclease rate, and therefore, excision of the mismatched base is greatly favored over polymerization on top of the mismatch. After this potential mutation is corrected, the primer template junction slides back into the DNA polymerase site, and replication continues.
And this is how the replication is going to be continued, and then it is going to take place, and the DNA is going to be replicated.
How well have you learned the mechanism of DNA synthesis and the functions of DNA polymerase?
So let us try to answer the questions based on whatever we have learned still in this particular lecture. So let us look at the questions which are provided to us. Okay. So question number 1 on your screen.
In this section, you will find out.
Answer the questions along with me.
Question 1. Which domain of the DNA polymerase contains most of the active site for DNA synthesis?
Everybody knows the answer, that it is always, which domain is going to be the one? It is always going to be the palm domain that is going to have the active site for the DNA polymerization to take place. So the answer is always the palm domain. Okay, it is this palm domain.
Correct.
Okay, moving forward to the next question.
Question 2. Which part of an RNTP is used by the DNA polymerase to discriminate between RNTPs and DNTPs?
So how does the DNA polymerase, such as Kornberg polymerase or the DNA polymerase 1, is going to differentiate between the ribonucleotide triphosphate and deoxyribonucleotide triphosphate? Please try to answer the question. I want the answer from everyone to actively participate in answering this question, whether it is 2' or it is 3'. Which is the hydroxy group that is going to be used for differentiating in between ribonucleotide triphosphate and deoxyribonucleotide triphosphate? Can you just differentiate in between the two? So it is always the 2' hydroxy group that is going to be used as a differentiating agent in between the ribonucleotide triphosphate and the deoxyribonucleotide triphosphate.
Incorrect. You may want.
Okay, sorry, this would be the 2 would be here because it is the first, second, third, fourth, and this is the 5th carbon atom which is present. Here it is the 2nd carbon atom. So that is why this is the 2nd hydroxy group that is present. Let me also label it out. So these are the labels. This is first, this is second, this is third, this is fourth, and this is 5th. So it is the 2nd carbon atom. Okay, checking the answer and moving forward.
Correct.
Okay, moving to the next question. See this hydroxy group is going to cause the excision of the ribonucleotide triphosphate to take place.
Question 3. Which phosphoryl group of the incoming DNTP is attacked by the 3' hydroxyl of the primer strand?
Okay, so the 3' hydroxyl group is going to attack which phosphate molecule? It is going to attack the alpha phosphate, the beta phosphate, or the gamma phosphate. So there are 3 phosphates which are present, and these are being designated here alpha, beta, and gamma. So it is always that the incoming nucleotide is going to be the hydroxy group is going to get converted into the oxyanion O negative, and then it is going to attack towards the innermost alpha phosphate molecules. So alpha phosphates are going to be attacked. So let me just give the answer. It is the alpha phosphate.
Correct.
Moving forward. Good. At least some students tried to answer.
Which of the following are functions of the divalent metal ions in the binding pocket? More than one answer may be correct.
So now you will have to illustrate that which are the different functions of the divalent metal ion in the binding pocket. And there are multiple number of answers which are correct. So let me just explain this to you. Recording. So you can find that the divalent metal ion here helps to deprotonate the 3' hydroxyl group of the primer. Yes, it is going to remove the 3' hydroxyl group of the primer. Here, this hydroxyl group is going to be, this hydrogen could be removed with the help of the metal ion. So the first option seems to be correct. Okay, so the first option is going to be correct. Attack the innermost alpha phosphoryl group. The attack on innermost alpha phosphoryl group is the function of the oxyanion and not of the metal ion. Metal ion का function ही oxyanion का function है. So B is wrong. Discriminate between ribonucleotide triphosphate and deoxyribonucleotide triphosphate. No, the difference between ribonucleotide triphosphate and deoxyribonucleotide triphosphate cannot be performed by the metal ion. Coordination of negative charge on pyrophosphate. Yes, coordination on the negative charge is being catalyzed by the metal ion. Holding base stacking interaction. No, base stacking interaction is being performed with the help of the tyrosine residue.
Correct.
So moving forward, see, the protons are being removed by the metal ion. Then the negative charge on the phosphodiester on the pyrophosphate molecules is also being neutralized with the help of the metal ions. Moving forward to the next question.
Question 5. Which of the following are functions of the finger domain? More than one answer may be correct.
Now, what is the function of the finger domain? Now, when you look at the finger domain, can you tell me what are the different functions? Helps to deprotonate the 3' hydroxy group of the primer? No, it is not the function of the finger domain. Holding the base in place through base stacking interaction. Yes, it is the function of the finger domain because finger domain has tyrosine residue which can hold the incoming nitrogenous base in their correct location. आने वाले नाइट्रोजनस बेसिस को करेक्ट लोकेशन में रखने के लिए फिंगर डोमेन के पास में क्या होता है? टायरोसिन रेजिड्यू होता है जो उन्हें करेक्ट लोकेशन पे रखता है. स्टेबलाइजेशन ऑफ द पायरोफॉस्फेट थ्रू लाइसिन एंड आर्जिनिन रेजिड्यू. यस, there are basic amino acid residue present over the finger domain, and these basic amino acid residue helps to keep the DNA at its own, keep the incoming nucleotides at its perfect position. Repair of mismatch base pair नहीं, ये काम नहीं करता. Introduces 90-degree turn in the template. Yes, it is always there because of this finger domain that an introduction of a 90-degree turn is going to take place in the DNA molecule. Now we will have to check the answer.
Correct.
Okay, so these are the three options which are correct. So you can look here.
Question 6. Which domain of the DNA polymerase is responsible for proofreading of the DNA chain?
So which of the following domain of the DNA polymerase is responsible for the proofreading activity of the DNA chain? So you can look here. It is the palm domain that performs the proofreading activity in the case of DNA polymerization reaction.
Correct. Question 7. How is the DNA polymerase able to remove mismatched base pairs while keeping correct base pairs? More than one answer may be correct.
Okay, so how does the DNA polymerase is able to remove the mismatched base pair? How does the mismatching is going to be removed? Replication is speed up when the mismatched base pairs are added? No. Replication slows when mismatched base pair are added? Yes. Replication is being slowed down when the mismatched base pair are being added. The exonuclease rate is faster than the replication rate when a mismatched base pair is present. Yes, the exonuclease rate becomes faster. The exonuclease rate is slower. The exonuclease site is able to specifically recognize mismatched base pair. Yes, it is able to recognize mismatched base pair, and those mismatched base pair are going to be removed. So this is what it is going to be supposed, and then we have to check it out.
Incorrect. You may want to.
Okay, so let us just look here. So replication slows when mismatched base pair are added. The exonuclease rate is faster than the replication rate when a mismatched base pair is present. No, the exonuclease site is able to specifically recognize the mis.
Question 6. Which domain of the DNA? Question 7. How is the DNA polymerase able to remove mismatched base pairs while keeping correct base pairs? More than one answer may be correct.
So now we will have to answer that how the DNA polymerase is able to remove the mismatched base pair. So the mismatched base pair is being removed with the help of an exonuclease site. As such, exonuclease site will not be able to differentiate between the correct nucleotides or the incorrect nucleotide. It is the palm domain, which has a site for recognizing the minor groove. Once the minor groove is going to be recognized, then the replication rate is going to be slowed down. Once the replication rate is going to be slowed down, then the exonuclease rate is going to be enhanced. And when the exonuclease rate becomes greater than the replication rate, then the excision of the nucleotides takes place. So what we find here is that replication is speed up when the mismatched base pair is added. No, replication in fact slows down when mismatched base pairs are added. The exonuclease rate is faster than the replication rate when a mismatched base pair is present. The exonuclease rate is slower. No, it is not possible. The exonuclease site is able to specifically recognize base pair. No, it never specifically recognizes base pair.
Correct.
Moving forward.
DNA synthesis requires.
So now we have moved towards the conclusion of this particular topic. So just have a quick revision about the same. So let us just revise it.
Deoxynucleoside triphosphates and a primer template junction. The 3' hydroxyl.
It is the primer which is providing the 3' hydroxyl group, and the incoming nucleotides are going to bind to it.
Of the primer strand attacks the alpha phosphoryl group of the incoming DNTP. Additional free energy is provided by the rapid hydrolysis of the pyrophosphate leaving group.
So the attack takes place by the oxyanion on the alpha phosphate molecules, the additional energy provided by the hydrolysis of pyrophosphate molecules. Next.
The main enzyme of DNA synthesis is DNA polymerase. Due to its shape, 3 domains of the polymerase, called the palm, fingers, and thumb, have been described.
So there are 3 different domains, palm, finger, and thumb.
The palm domain houses the active site for DNA synthesis. The palm domain can distinguish between RNTPs and DNTPs. 2 divalent metal ions in the palm domain are crucial for DNA polymerization activity.
Okay, so there are 2 metal ions which are involved in this process. Next.
When a correct base pair is formed between the incoming DNTP and the template, the finger domain moves to enclose the DNTP. This conformational change brings the divalent metal ions into the correct position to function. Lysine and arginine residues on the finger domain help to stabilize the pyrophosphate, and a tyrosine residue helps to hold the DNTP in place for catalysis.
Okay, once the tyrosine residue comes in place, it is going to keep the base stacking interaction in place and keep the nitrogenous base in its exact location. The lysine and arginine residue minimize the negative charges over the phosphate molecules, the beta and the gamma phosphate.
The finger domain also associates with the template region, leading to a nearly 90-degree turn of the phosphodiester backbone of the template strand. The thumb domain interacts with recently synthesized DNA, serving to maintain the correct position of the primer template junction and reduce the rate of dissociation of the polymerase from the substrate.
So the finger domain, finger domain just attaches the DNA and keep it in its perfect location.
Another function of the palm domain is proofreading. When a mismatched base is added, the replication rate slows dramatically, and the primer template junction is free to interact with the exonuclease site.
Okay,
the exonuclease site.
This is the exonuclease site which is present on the palm domain. The exonuclease site then cleaves the last added nucleotide triphosphate, and the error is being removed.
Removes the mismatched base, and replication continues.
So this is how replication is going to take place.
You have completed this exercise.
With this, we have finished this particular process. The last diagram indicate the summary of what we have studied in the case of DNA polymerase 1. That's all for today. Hope you have enjoyed the lecture. If there is any query, you just write it down, provide it on my WhatsApp or on the chat box or on the comment.